how do you and Bayer plan to work together?
Yeah, we're really excited about this collaboration that we announced today because at Mammoth, we've built out this incredibly broad toolbox of novel CRISPR systems, including ultra small CRISPR proteins that can enable new ways of doing in vivo delivery. And this collaboration with Bayer is all about expanding their cell and gene therapy platform with these novel CRISPR proteins that can really enable especially these in vivo therapies that are exciting.
You're focused on 4 diseases, all liver-based diseases. Why those diseases, and where do you hope to expand?
So liver is the starting point. And in addition to kind of starting with liver, I think that the potential for these novel proteins that we're working on at Mammoth for these in vivo applications across many different organs is limitless. I think it has a lot of really exciting directions that it can go, but liver is a great place to start, and that's where we're starting with Bayer. But broadly, we're excited about the internal work we're doing at Mammoth. We're excited about the work with Bayer, and I think we'll see the power of these ultra small CRISPR systems in a variety of tissues.
Now, obviously, gene editing technology has been incredibly controversial. What is your company's approach to the ethics of gene editing? It's certainly one thing to cure someone of sickle cell anemia, but it is another to be able to choose the color of your baby's eyes or height and beyond.
So at Mammoth, we're really focused on these cures for really debilitating genetic diseases. And there's so many of these that we could have such a high impact on with patients that could be our focus for decades and hundreds of years and still not meet the full need. So I think that's what we're really, really focused on. And I think ethically, personally, that's what you have to focus on is, right? There's people that really could be cured permanently of these diseases if we can apply this technology in the right way.
What's been the impact of the pandemic on the gene editing sphere? Obviously, we know, how important mRNA technology has been. Talk to us about how the pandemic might be a turning point for gene editing technology.
Well, one of the interesting things is in the development of our CRISPR-based platform, one of the new uses of CRISPR that we've pioneered is actually diagnostic applications. And the pandemic has really accelerated our development of those technologies in that area. And you may have seen previously, we've announced collaborations and partnerships with GSK, the NIH, and DARPA. And I think really it's shown that CRISPR is something that can go toe-to-toe with gold standard technologies in the diagnostic space like PCR, and really offers the potential of kind of revolutionizing the whole spectrum of testing for infectious diseases, starting with COVID-19 and beyond, whether that's in the lab or that's in the doctor's office or even at home.
Meantime, we just got a historic verdict in the case of Elizabeth Holmes, as given that she was a young, then up-and-coming CEO and founder in the biosciences space. I'm curious what impact she's had on the work that you do and the work you're trying to do, and if you have any thoughts on it.
Sure. So at Mammoth, first and foremost, we like to lead with the science, and we've published many papers in top scientific journals, and we're very excited to explain to people how we use CRISPR across therapeutics and diagnostics. And I think it really just reinforces the importance of really just being transparent and open and getting people excited about how the technology actually works and helping them understand what it can do and what it can't do. I think it just really reinforces that for us.